A child is born looking entirely normal. Within a year, something has quietly shifted inside every cell in her body, and the consequences will become visible to anyone who sees her. Progeria, formerly known as Hutchinson-Gilford Progeria Syndrome, or HGPS, accelerates the physical markers of aging at a rate roughly 8 times faster than typical human development. Children who carry this condition do not lose their minds or their spirit. Their mental and motor function remain intact. What changes is their body, and it changes relentlessly. To understand what that means, you first have to understand something about how the human cell holds itself together. Inside each of our cells sits a nucleus, the structure that contains our genetic information. That nucleus needs a kind of internal scaffolding to keep its shape and allow normal cell division to happen. One of the proteins that builds that scaffolding is called Lamin A, and it is produced by a gene called LMNA. In children with progeria, a single-point mutation, one letter changed in one gene, causes the body to produce a defective version of Lamin A. Researchers name this faulty protein progerin. Progerin does not behave the way Lamin A should. Instead of supporting the nucleus properly, it destabilizes it. The cell's inner nuclear envelope becomes misshapen. DNA repair becomes impaired. Chromosomal instability increases. And because this is happening inside virtually every cell, the damage is not isolated. It accumulates across the entire body over time. Most children with progeria appear completely normal at birth and through early infancy. The first warning signs typically emerge in the first few months of life. A physician might note a failure to thrive, meaning the infant is not gaining weight and growing at the expected rate. There may be an early skin change, a localized hardening and tightening that resembles a condition called scleroderma. These early signs are subtle enough that they can be missed or attributed to other causes entirely. By around 18 to 24 months, the picture becomes harder to misread. Growth slows markedly. Hair loss progresses to full-body alopecia, complete absence of hair across the entire body. A distinctive facial structure develops, a small face relative to the head, a shallow recessed jaw, a pinched nose, and prominent eyes. Scalp veins become visible beneath the skin. The body remains small and fragile-looking. The proportions carry an unmistakable resemblance to extreme old age in a child who has barely learned to walk. This is the contradiction that sits at the center of every progeria case. The child's mind is present, engaged, and developing along a typical timeline. But the body that carries that mind is aging at a pace no treatment could halt, at least not until relatively recently. As these children move through early childhood, the musculoskeletal system deteriorates further. Body fat disappears, muscle mass reduces, joints stiffen, hip dislocations occur, skin across the trunk and limbs hardens. And underneath all of that, in the circulatory system, the most dangerous process is quietly accelerating. Atherosclerosis, the narrowing and hardening of arteries caused by the buildup of fatty plaques, is a condition most commonly associated with elderly adults and decades of accumulated cardiovascular stress. In children with progeria, it develops well before puberty. The cardiovascular complications this produces are the primary cause of death in nearly all documented cases. Most individuals with progeria live into their mid to late teens or early 20s. Severe cardiovascular disease typically develops by the time they reach adolescence. The mechanism that connects the single LMNA mutation to this cardiovascular outcome took years to work out. Here is what the evidence shows. When progerin accumulates inside cells, it interferes with the normal processing of Lamin A, the precursor molecule that should eventually become functional Lamin A. Under normal conditions, Lamin A goes through a carefully sequenced series of enzymatic modifications before a protein called ZMP-STE24 cleaves off its final 15 amino acids, producing the stable, functional form. When the mutation is present, that final cleavage step is blocked. The intermediate form, still carrying a chemical tag called a farnesyl group, becomes trapped. This trapped, farnesylated progerin embeds in the nuclear membrane and prevents it from functioning normally. The result is a cell that cannot divide cleanly, cannot repair its DNA reliably, and accumulates damage faster than a healthy cell would. Multiply that across billions of cells, across years of development, and the organ systems most sensitive to cellular turnover, particularly the cardiovascular system, begin to fail on a timetable that mirrors, in accelerated form, what happens in ordinary aging over 7 or 8 decades. This is where the scientific importance of progeria extends beyond the children who carry it. Because progeria is caused by a sporadic mutation, meaning it arises newly in the affected individual, not inherited from parents, it is extraordinarily rare. Estimates suggest it affects roughly 1 in every 4 million newborns. The small number of affected individuals worldwide has made clinical research difficult, but the mechanism itself has become a lens through which researchers examine normal aging. Progerin, the defective protein, has been detected in small amounts in the cells of typical older adults. This observation has led researchers to ask whether the same pathway that drives catastrophic early aging in progeria might play a contributing role, far more slowly, at far lower levels, in ordinary cardiovascular aging. That question remains open. The detection of progerin in aging cells is documented. Whether it contributes meaningfully to typical age-related cardiovascular disease is a hypothesis that the current evidence has not resolved. What the evidence has resolved is the causal chain within HGPS itself. In 2003, researchers identified the precise point mutation responsible, a substitution of cytosine for thymine at position 1824 of the LMNA gene. That discovery, more than any previous research on the condition, gave scientists a defined molecular target. It changed progeria from a described syndrome with no known mechanism into a condition with a specific, understood fault that could, in principle, be addressed. For families navigating this diagnosis, the molecular explanation does not change the daily reality of a child whose body is aging beyond what any family anticipated. What changes is the possibility, still partial, still limited, that understanding the mechanism well enough might one day alter the trajectory. Children with progeria retain their intelligence, their personality, and their capacity to form relationships throughout their lives. The disease does not take those. What it takes, systematically and without pause, is physical resilience. The stiff joints, the skin changes, the loss of body fat, the progressive cardiovascular disease, these are not background details. They are the daily experience of children who understand, at some level, what their diagnosis means. The story of progeria is not a story medicine has fully resolved. Progerin is understood. The mutation is mapped. The nuclear mechanism is documented. But the children who carry this condition still face a foreshortened life, and the gap between knowing the mechanism and correcting it remains significant. The research that progeria has generated about Lamin A, about nucleus stability, about the biochemistry of cellular aging represents a body of knowledge that would not exist without the individuals who were diagnosed, studied, and whose lives gave researchers something concrete to investigate. That is the real contribution of every documented progeria case, not the disease itself, but the knowledge extracted from it with care, with the consent of families who understood what participation might mean for children they would never meet. This video is based on publicly documented medical and scientific sources and is intended for education and storytelling only. It is not medical advice. If you or someone you know is experiencing concerning symptoms, please consult a licensed medical professional. The question progeria leaves open. Whether the same molecular pathway that collapses in these children operates quietly in all of us as we age is one the biology is not yet finished answering. That question is where the research is going now. Medical mysteries. Every symptom has a story. Every patient deserves to be heard. Medical disclaimer. This video is based on real, publicly documented medical case reports and is intended for educational and storytelling purposes only. It is not medical advice. If you or someone you know is experiencing similar symptoms, please consult a licensed physician immediately.